Hello, my name is Will Cito. I'm a junior at St. Paul Academy, and this year I've been researching the algae Dydymasphenia geminata. I'm first going to give a little background on the organism, then talk about my research process, what conclusions I was able to reach, and finally how we can apply my research to the real world. Now, in Vestas didymo, this diadem algae is commonly found in cool, fast, oligotrophic waters. There are a small number of actually living cells like seen on the right, as most of its mass is made up by the dead carbohydrate and phosphorus-rich stalks you can see on the left, from which its other name of rock snot comes. Didymo is distinguished from similar diadems in part by how it is able to connect enough nutrients from oligotrophic water to produce a thick layer of stalks that can smother an entire river for miles. In the past 30 years or so, didymo has become a nuisance species across much of the world. Its historical range is contained within North America and Europe, but in recent years it has spread as far as Chile, Argentina, and even made the jump to New Zealand. But more than simple geographical spread, researchers have found it increasing in its density and prevalence where it has existed in small numbers for years, with no clear answer as to why this is happening now. And its density is quickly becoming a problem, too. Large didymo blooms can disrupt natural cycles and other organisms in the river, and can even impair human enjoyment. Smaller amounts of the algae's snot-like appearance have been at times reported to local water treatment plants as a sewage leak, helping the algae live up to its rock snot name. Its impacts are already beginning to be seen financially in nature-based tourism, like the nearly $1 billion fly fishing industry, and many governmental bodies have begun treating didymo as an invasive species. Despite the fact that a not insignificant amount of research has been done into didymo, we still don't know what exactly is causing its spread and proliferation, and this lack of concrete conclusions is due in large part to the fact that didymo has yet to be grown in a laboratory environment. Scientists have had some success culturing individual cells, but only being able to work with didymo blooms in the field has complicated any potential research. So I decided to research how we can grow didymo in a lab. To best emulate its natural growth environment, I identified the 8 chemical and physical variables which seem most relevant from my background research and took a deep dive into the raw data from both studies and USGS river monitoring records. In all, I ended up consulting over 30 datasets and research studies with more than 200 total data collection sites represented. The process differed slightly for each variable, but generally I tried to identify patterns I found in the data in literature and verify those with more data. So let's dive into the variables. The general profile of a didymo-infested water body describes a simple rocky substrate like pebbles or cobbles, as confirmed in all available research. In an interesting study, West et al. 2020 found that didymo cells would return to rocks they had been scraped off of by researchers, but would not bloom as commonly as on rock didymo had colonized for longer. Other studies have not always agreed with this, but generally a substrate from where the didymo sample has been collected would be recommended for any growth. Integral to stalk building is sunlight, or more specifically, photosynthetically active radiation. This variable was very difficult to find numerical data on, as how much light actually penetrates to the cells is incredibly site-specific. An assortment of studies from Drs. Kilroy and Bothwell have shown the bloom prevalence, stalk production, cell attachment, and cell length of didymo are all favored under higher light. Similarly, my own data analysis shows the most successful didymo growth in 5 to 15% tree canopy coverage areas. Any artificial light source used must also contain ultraviolet radiation, as studies have found similar stalk diadem algae growing 2 to 4 times greater abundance under UVR. As a related aside, I think further research is warranted in the absolutely fascinating hypothesis that the recent global increase in UVR has influenced the recent global increase in didymo. This is one example of research that is most possible in the lab, not the field. Didymo cells have been collected from a range of temperatures, but most commonly from cold water. From my data analysis, the species is generally found in a summer water temperature of around 15 degrees Celsius, and detailed data from USGS sites show temperatures reaching around 0 degrees Celsius in late May and rebounding throughout the summer. Cell growth has been shown to continue even in winter, but no research has identified how this temperature cycle affects didymo and what the implications of a change in climate may be for the species' continued expansion. I was unable to find any numerical data for water velocity, but all available research described didymo as preferring fast-moving turbulent water with a relatively constant flow, like where it is commonly found below dams. A specific speed may have to be discovered through in-lab adjustments. Out of all the variables I studied, phosphorus is perhaps the single most researched. It is central to what scientists have deemed the quote-unquote Dymo Paradox, the fact that didymo stalks are made from soluble reactive phosphorus, or orthophosphates, yet phosphorus concentration is negatively correlated to didymo presence. In their extreme literature and field study of didymo and orthophosphates, West et al. 2020 found that the most common threshold for growth rate was very low. In South America, no blooms were observed above 4 micrograms of orthophosphate per liter, in New Zealand rarely above 3, and in the American Rockies rarely above 2. Kilroy and Larn in 2016 identified a potential threshold of around 2 micrograms of orthophosphate per liter, a threshold that appears again in Coons et al. 2018 and James et al. 2015. A suggested value of 2 micrograms seems reasonable, but this is likely a variable that will be manipulated frequently. In situ studies from 2011 and 2014, Bothwell and Kilroy found that nitrogen was not a primary limiting nutrient in Dymo growth. Another study created nitrogen concentrations as high as 250 and as low as 10 milligrams per liter, yet found no detectable effect. A proposed bottom threshold of 10 milligrams per liter has been suggested, below which cell division and stock development is limited, but above which growth seems to continue largely unimpeded. Every applicable measurement I had found nitrogen values that fell within this proposed range of 10 to 100 milligrams, with the most clustering around 40 to 50. Iron levels, the focus of multiple papers, has at times become controversial within the scientific community. It was first hypothesized in Sundaresh 2011 that Dymo growth was in part predicated on the availability of highly reactive iron after they experimentally found this connection with dead Dymo stocks. This result was emphatically disputed in studies like Bothwell 2012, who combined a literature and field-based approach to conclude that Dymo occurs best in low iron water. This was mirrored in James 2014, who found iron levels to be inversely related to Dymo bloom size in 39 sites along one river. The median iron values for locations with confirmed blooms in one study ranged from 0.05 to 0.20 milligrams of iron per liter, but clustered around 0.08. Out of 7 studies and databases I analyzed that focused on iron, the data of 6 fell within this projected range. And our final of the 8 conditions, pH. Dymo has been recorded in a variety of pH values, from as low as 6.7 to as high as 9.62. A lot of variation occurs within this range, and again, this is a variable we simply don't yet know enough about. This table represents a summarization of the results I detailed in the previous slides. Although many questions remain as to what these factors actually determine the success of Dymo, all available data and research seems to agree on this relatively narrow window for the best growth. So, where do we go from here? The next significant step in this research would be trying out these hypothesized conditions to verify that they do, in fact, work. This is a project I'm planning to take on this spring with the assistance of some of the scientific professionals I connected with during my research process. A proposed setup would include a long, wide, deep artificial riverbed in a style similar to the wave tank pictured, with cobbles and water collected from the same river as the Dymo sample. Conditions would be checked frequently to verify nutrient levels, and water could be rapidly recirculated through the tank to maintain speed. Water would enter the tank with a significant drop, similar to that seen on the Libby Dam, under which Dymo currently lives, to help create those preferred turbulent conditions. The successful growth of Dymo in a lab will open a world of possibilities. As I see it, science stemming from this work falls into 3 main categories: mitigation, scientific understanding, and technological applications. First, mitigation. While in situ testing will obviously be needed too, Dymo blooms in a lab can be experimented on without fear of permanent impacts to the surrounding environment. Any mitigation strategies that were developed to control Dymo blooms would be rapidly embraced by regulatory agencies and outdoors groups hoping to enjoy our rivers for generations to come. Second, scientific understanding. Observing Dymo's nutrient intake up close can help us better understand its processes and can begin to present solutions to questions like the aforementioned Dymo Paradox of remarkable phosphorus uptake. In my research, I have been forced to focus on correlation, but this would allow us to study causation in all of these variables. For example, does Dymo legitimately prefer cold water, or does the actual controlling variable just happen to always occur alongside cold water? Does the hypothesis about UVR causing Dymo expansion hold any water, if you will? We need to learn more about this vexing and important species. And finally, technological applications. Humans across history have borrowed so much of our smartest technology from other organisms' developments, and I suspect Dymo may be no different. It's hard to know where exactly science will lead us, but one of the more specific examples lies in phosphorus reclamation. Dymo is famous for efficiently harvesting phosphorus. Once we understand how exactly it does that, can we use the same technology to remove phosphorus from rivers dangerously polluted by phosphorus-based fertilizer? Can we repurpose these dead phosphorus and carbohydrate-rich stock materials as fertilizer itself? Truly, so much is possible here, but it is all predicated on being able to study Dymo more closely in a lab. I hope my work is able to help propel us towards that possibility. And lastly, here are my resources. Thank you so much for listening to my project, and I hope I will get a chance to answer any further questions you may have.